from New York University. Michael, of course, is a longtime friend of the Barth Syndrome Foundation and has made some seminal discoveries in the molecular mechanism of tafazzin, and today he's going to talk about the mechanism of acyl-specific phospholipid remodeling by tafazzin. Michael.
Okay, thanks. And so if I'm following Steve's logic, then I'm the biochemist who does not remember the question. And you will see that sometimes it's useful not to remember the question. And perhaps I can give a partial answer to the last question that you asked, but we'll see whether you buy it or not. So we've continued to be interested in tafazzin, and here's just to remind everybody of a tafazzin reaction. That's an acyl or a trans-acylase that transfers an acyl group from a phospholipid to a lysophospholipid, producing another lysophospholipid and another phospholipid. The reason why we've continued to be interested in that is because there are still some lingering questions which are actually very central, namely revolving around the acyl specificity of that enzyme as well as its general function, and that's really not understood. And that is, at least as far as I'm concerned, at the heart of the pathomechanism of Barth syndrome. So the study I want to, or the data I want to share with you are all new data, and they came out of two unexpected observations. The first unexpected observation was made in mitochondria, isolated mitochondria, where we measured the amount of tafazzin by quantitative Western blot using fluorescence-labeled antibodies. And then we also measured the amount of phospholipid that react with tafazzin, and that was done by taking isolated mitochondria, incubating them with lyso-PC, radiolabeled lyso-PC, and we waited till the reaction reached equilibrium, and then we back-calculated the amount of phospholipid that had reacted with tafazzin from the radioactivity distribution of LPC and PC. And it's important here to note that in the equilibrium state, the LPC, which is the substrate that we are supplying, was not depleted. So this is not what's limiting the reaction. But if we then calculate the number of molecules of phospholipids that reacted per molecule of tafazzin, then the number in wild-type fly mitochondria is below 1,000. Which then, if you compare that to the total amount of mitochondria and lipids, it's less than 1%. So tafazzin reacts with less than 1% of mitochondrial phospholipids. So then we overexpress tafazzin, and of course the tafazzin concentration goes up as expected. The amount of phospholipids that react with tafazzin also goes up, but the ratio actually goes down, suggesting that now the amount of trans-acylation competent phospholipids in mitochondria is the limiting factor. So from this we concluded that tafazzin reacts only with a tiny, tiny portion of mitochondrial phospholipids. And therefore, a barrier must exist that prevents the widespread trans-acylation of the entire membrane. And so we wanted to understand what this barrier is. So to study the physical and chemical properties of tafazzin, we sort of went back to a very, very simple reductionist incubation system that consists only of purified tafazzin and a lipid mixture that typically forms liposomes. And we demonstrated that tafazzin binds to these liposomes very tightly. And here's where we made our second unexpected observation. And that is a look at this reaction here. This is quite familiar to you if you've looked at our trans-acylations assay. So we made a liposome consisting of one palmitoyl linoleoyl PC and radiolabeled palmitoyl lyso-PC. And if you look at the trans-acylation reaction, the products and the substrates and the products are chemically identical, right? The only thing that happens is that the radioactivity moves from LPC to PC, and we can measure that the enzyme actually does catalyze the reactions. The enzyme is active. But since there is no thermodynamic gradient between substrates and products on account of, based on the fact that they are actually identical, you would expect that the radioactivity in equilibrium now distributes according to the PC-LPC ratio, right? Which is 20 to 1. But we only found 1 to 45. no matter what we do. So here we have a situation where tafazzin is active. It does react with phospholipids. We can clearly show this, but it reacts only with very few phospholipids, and then it stops. Right? So this was very depressing initially, and then eventually I got myself together to follow up on this more carefully, so we spun down these liposomes, and of course you can actually see them with your naked eye. 100% of these liposomes come down after ultracentrifugation. But about 80% of the radiolabeled PC remains in the supernatant. So this is when I realized one of our assumptions wasn't right. And we had, of course, assumed that the lipid mixture that we were forming forms liposomes. And this is, of course, the case because we can see the liposomes. And in a PC, LPC 20-to-1 mixture, you expect liposomes, and there are liposomes. You can actually see them also by electron microscopy, which we did. But, of course, it doesn't exclude that they also form micelles. A small fraction of this, right? That's a possibility. So our hypothesis here was, at this point, that tafazzin binds to liposome, but doesn't react with liposomes. It only reacts with micelles. Okay, so now we followed up on this, and then we moved away from these radiolabel studies and moved to mass spectrometry because it allowed us to make lipid mixtures of variable concentrations without worrying about using too much radioactivity. So in this experiment, we looked at different PC-LPC mixtures. Each mixture contained 100 nanomole total lipids, and when we varied the LPC-PC ratio from 0 to 100 to 100 to 0, right? So at the extreme ends, you see no reaction, of course, because there's only one substrate. In the middle, there is reaction, as you would expect. But if you look at this more carefully, you see there's one big peak here. This is when the mixture is dominated by LPC, and there's virtually no reaction here, and the mixture is dominated by PC. And we did magic angle spinning NMR to find out about the actual phase state. And as expected, we find this to be in the micellar state and this to be in the bilayer state. And we concluded here that in the PC-LPC system, tafazzin does not react with lipids when we are on the bilayer state, but it does react with lipids when we are on the micellar state. So now what about other phospholipids? Like, for instance, phosphodietylethanol or cardiolipine. These are lipids that do not necessarily have to be in the bilayer state. As we all know, they can form various other phase states. And we did very similar experiments, and just to sort of make this simple, I've summarized these experiments here for you. You can see that the red symbols here, they indicate the tafazzin activity or the transisolation activity. And these squares, they indicate the phase state. And the phase state was determined by magic angle spinning NMR. So just to sort of follow the way I'm depicting it here, the light blue means the aqueous phase and black means the lipid phase. Right? So this is the bilayer phase, and this is something with positive curvature. That's a micelle. Tafazzin is a membrane protein. It binds to membranes, so it's hydrophobic. It has also hydrophilic sites. So the way we isolate it is in the micelle. So it comes in a Triton micelle. And as I said, it binds to the liposomes. Excuse me. So just again, in the PC-LPC system, things are very simple. No reaction in the bilayer phase, but a lot of reaction in the micellar phase. Now, in the PE-LPC system, it's becoming more interesting because here, if the system is dominated by BE, we're having an inverted hexagonal phase. Tafazzin is active in that phase. We can also form a micellar phase, as LPC becomes the dominant component, and tafazzin is active as well. Most interesting is the cardiolipin LPC mixture, because in cardiolipin LPC mixtures, we have coexistence of two phases, the primary phase is the major phase is in the bilayer phase. It's in bilayer configuration, while a small portion is in an isotropic phase, which according to our cryo-EM and NMR studies is probably micellar, although we are not 100% sure about that. So there is basically a bilayer phase and a small portion of a non-bilayer phase, and this is somewhat active. But luckily, in that mixture, you can convert the entire system into a hexagonal phase by adding calcium. And we did this, and this greatly accelerates the activity, the trans-acylation activity. And I will get into this at the end of the talk to show you the actual data, because this is, of course, the most important part. So we are concluding here, tafazzin does not react with lipids when we are presented as bilayers. It only reacts with lipids when we are presented in some sort of non-bilayer phase. And the possibilities here are, of course, there is more than one bilayer phase that works. So what is the common denominator of non-bilayer phase? It's curvature. They are curved. So why is it important for lipids to be in a non-bilayer phase in order to react with tafazzin? And this remains speculative, of course, at this point, but I will show you some interesting data which I think give us an idea of what might be behind that. So we looked into a series of PC-LPC mice cells, and we used different molecular species varying the number of double bonds on the acyl chains as well as the chain length. And you can see here that the trans-acylation activity increases with the number of double bonds and decreases with the chain length. And whenever you have a phenomenon where the activity increases with the number of double bonds and decreases with the chain length, that is related to acyl chain packing, or what is sometimes also called fluidity, right? So why is it that fluidity might be important for the tafazzin reaction? So my best guess is that fluidity will enable ideal mixing of lipids and lysophospholipids. And that is something I sort of gradually learned while I was doing these studies, because I've always assumed that lysophospholipids, at least in small amounts, can easily dissolve in a bilayer and just sort of float around there and don't have any particular behavior. But that doesn't seem to be the case, as you can see in the next experiment. So here, this is sort of the simplest possible trans-acylation system you could think of. You have LPC, 14-0, and you have 14-0, 14-0 PC, except that the 14-0 chains of the PC are deuterium labeled. And the trans-acylation product that you expect is a PC that contains a labeled and an unlabeled myristoyl group, and that you can, of course, easily identify by mass spectrometry. So what's important here is that there is only 14-0 chains in the system. Again, it's critical because there's no thermodynamic gradient. You would expect that they being randomly shuffled around, right, by tafazzin, if tafazzin is active. If tafazzin is active, you can actually follow its activity over time. The reaction ceases or reaches its equilibrium at about 40, 50 minutes, and then nothing else happens anymore. And then again, this is sort of a recurrent theme. If you back calculate the amount of acyl chains that have been transferred, it doesn't add up. Only a tiny, tiny portion of acyl groups have been transferred at that time, which can only be explained by segregation. So if you mix LPC, 14-0 LPC and 14-0, 14-0 PC, they do not mix. Right? That's something that escapes, of course, our eye. We mix them together. It's some sort of a clear solution because LPC is a dominant lipid. It looks like a micelle. If you shake it, it will form. You think, oh, everything is nice and dandy. But probably what's happening here is that the PC segregates in small, liposome-like lipid assemblies, and the LPC stays separate. So the enzyme never sees the two substrates together, right? So we are concluding here that tafazzin does not react with lipids in the bilayer state. Instead, it requires substrates to form assemblies with high positive or negative curvature, such as micelles or inverted hexagonal structures. And low lipid packing order promotes trans-acylation, presumably because it facilitates ideal mixing of phospholipids and lysophospholipids. And just to connect this now, not to forget the question completely, to mitochondria. Of course, mitochondrial lipids are predominantly in the bilayer state. That has been very clearly shown. But there are some portions in mitochondria where the lipids cannot be in the bilayer state. That's physically impossible. For instance, here, the curvature is simply too high. No bilayer will survive that, right? Or obviously, if membranes fizz or fuse, where you have always non-bilayer intermediate, there's no other way to do that. So it is possible, although it has never been shown directly, that mitochondria contain non-bilayer structures. So now the rest of the time I want to use to show you perhaps the even more relevant result, and that is that of acyl specificity. So we have shown that tafazzin can react with virtually any phospholipid and lysophospholipid that we have tried. And so we're having now something like 10 to the 3 phospholipids and something like 10 to the 2 lysophospholipids, which gives us 10 to the 5 different transacylation reactions. So which raises the question, what is the purpose of having such an array of transacylation reactions? Why is it that tafazzin does not equilibrate the fatty acid composition of all phospholipids? Because this is what you would expect, right? And why is it that it actually creates acyl specificity in cardiolipin and perhaps in some other phospholipids? So the first experiment in which we addressed the question of acyl specificity is this one. And here we sort of we looked at a cardiolipin LPC system in which we let the transfer of oleoyl and linoleoyl groups of cardiolipin to compete with each other. Just to take you through the data, we saw multi-spectra. If you have 18-1 cardiolipin as your only substrate or only acyl donor, then and 19-0 LPC is the acyl acceptor in this case, but we've tried it with our ones too, so it works no matter what you use. But in that case, the product will be 19 or 18-1 PC. If you use 18-2 cardiolipin, obviously the product will be 19 or 18-2 PC, so the mass is shifted to the left by 2 units. And if you have a 1-to-1 mixture of 18-1 and 18-2 cardiolipin, the two products will appear about at a ratio of 1-to-1. Now you are adding calcium and converting the system into a hexagonal phase, and you can see everything else holds true, except here now in the competition experiment, we have exclusive acyl specificity for 18-2. There's no formation of 18-1 at all anymore, right? So, and that, encouraged by this result, we were now trying to reconstitute the situation of mitochondria in vitro using, well, I have to admit, a simplified mixture of mitochondrial lipids, because otherwise it would get too complicated. Specifically, we used cardiolipin and PC as our dominant phospholipids, and we threw in 2 lysophospholipids, namely monolysocardiolipin and LPC, at lower concentration. We actually modified these concentrations, and the results are very similar. So the acyl species were designed in such a way that the system enables 4 symmetric transacylations, namely the transfer of 18-2 from PC to monolysocardiolipin or from PC to LPC, and the transfer of 18-1 from cardiolipin to monolysocardiolipin or LPC. So we formed this mixture and we added tafazzin. And you can see that actually not much is happening. A little bit, we're seeing two transacylation products. Primarily, acyl groups are transferred from PC to LPC and from cardiolipin to LPC, but the activities is very low. Now, if you look at the NMR, you can see that this particular mixture is primarily in the bilayer phase, as you can see here by this low field shoulder. A small portion of it is also in an isotropic phase, which is probably a micelle, which is probably the one that is active here. Now, when we add calcium, we convert the system into a hexagonal phase, which is shown here in the mass, which is evident from the NMR spectrum by this high field shoulder that comes up here, right? And this is an almost complete conversion. There's very little that remains in the micellar phase. So after adding calcium and after converting that system into the hexagonal phase, we're seeing a drastic change in the trans-isolation pattern. First of all, the overall trans-isolation activity increases. We are seeing various products. All four reactions get activated. But most importantly, we are now seeing the infamous tetralinolenyl cardiolipin in relatively large concentration. So I also want to mention that calcium has no effect whatsoever on other trans-isolation reactions of tafazzin. It's a pure phase effect. The calcium does not act on the enzyme. We can show this. We have shown this in many other reactions, both with negatively charged phospholipids as well as with neutral phospholipids. But this experiment shows very clearly that we can reconstitute the mitochondrial situation in a very simple test system consisting only of 4 phospholipids and purified tafazzin if we do the reaction in the correct phase state. So we concluded that the acyl specificity of tafazzin depends on the lipid phase state, and native molecular species of cardiolipin can be created in vitro if tafazzin reacts with mitochondrial lipids in the hexagonal state. So what is it that connects tafazzin to lipid curvature, if you will? So this is sort of my final slide. And so this is, of course, a hypothesis, and I'm not saying that we have all the data to support that model, but I think it is at least plausible at the moment. And it tries to answer now not only the question how does this phenomenon of acyl specificity can be explained, but actually more importantly, why do we have tafazzin in the first place? What is it really good for? So this shows a lipid bilayer. Of course, I'm showing only one monolayer here just for the sake of clarity. The circles are the head groups, and then there's 2 types of fatty acids, namely blue ones that are saturated and yellow ones which are unsaturated. And you can see that most of these lipids are unsaturated, saturated, so they contain a saturated chain and an unsaturated chain, and they are perfectly aligned in a bilayer assembly. So everything is nice until here, until some external force comes in that bends the membrane, right? Because the membrane, if the membrane bends, and the membrane can bend in mitochondria for various reasons, this will exert significant stress on the packing of phospholipids. They will not like that. A phospholipid assembly that is okay in the bilayer state will not like to be in a non-bilayer state or any form of curved state. So this would this would resist that bending process, or it would mean that this bending process would have a very high energy burden. Now, if tafazzin is present, tafazzin can easily reshuffle all the fatty acids being the phospholipids and can create, for instance, di-unsaturated phospholipid species of which we know that they are cone-shaped. That is relatively well established for many of them, at least. Cone-shaped phospholipids will fit in nicely here, as you see. And now, again, we have ideal packing. So what tafazzin does, according to this hypothesis, is that it reduces the thermodynamic cost of membrane dynamics, membrane bending and membrane stretching. And of course, it is important to realize that this process is reversible, because this membrane that is bent now may have to be flat again at some point during its lifetime. And of course, the tafazzin reaction is reversible, and it can go to wherever the system wants to take it. So I want to acknowledge my collaborators. Of course, here my collaborators at NYU are well known in this group. I don't want to mention them specifically because they are all here. What I do want to mention is Richard Epent, and he and Bob Berner at McMaster's University. They have done the NMR study, so I've become a frequent visitor to to Hamilton. I met Richard 2 years ago here at the Gordon Conference. This is one of the many good results of these conferences. And the collaboration with Richard has been absolutely essential for this project. Thank you.
Michael, just a quick question. So can the calcium concentrations that you've shown, at least in vitro, to make this transition, are those achieved in vivo? Could they be achieved in vivo?
No, not at all, no. And I don't think, this is a pure in vitro system. So these 15, or sometimes we use 20 millimolar of calcium, don't exist in, the cell would be dead if they, But I'm not saying that you have this kind of hexagonal structures in the mitochondria. I'm not saying that at all. In the mitochondria, things, of course, are more subtle. But you will have non-bilayer structures at certain points in the mitochondria. And they won't require the presence of calcium. They will, our forces can create these kind of non-bilayer structures. And I think the tafazzin reactions takes only place in those non-bilayer structures. And of course, I think we will have to, now we are trying to identify what these non-bilayer structures are and where they are and what they actually are. They could be, the two main candidates are, it could be fusion intermediates, membrane fusion intermediates, that is one possibility. The other main possibility, it could be the cristate junctions, basically, because in the cristate junctions you have an enormous curvature. Most people don't realize that. Because the diameter is something like 20 nanometer. No liposome will be stable under these conditions.
Do you think de novo synthesis could drive this? De novo synthesis of cardiolipin could drive the formation of these structures? And then you get the rapid subsequent remodeling with the tafazzin?
We don't know, of course. If I have to speculate, I would say I would think of other candidates first for this, but obviously we don't know. Yeah.
I have a couple of questions. I don't remember what it was, but isn't there a precedent for some enzyme activity that occurs in the bends? I know I've read a paper that demonstrated that this reaction occurs only in the curved areas. Do you remember what that was? Have you read that paper? Yeah, so it's not unprecedented, in that
Remember,
sense.
it's about forgetting the question.
So what?
No, I'm joking. It's just about forgetting, yeah.
And the other question I have is, given the diversity of species of cardiolipin, is the structural hypothesis compatible with that diversity? That is, if structure is a major determinant, then there must be some correlation with structure in other mitochondria and other species, if I understand your hypothesis. So
is the hypothesis compatible with the presence of so many different species, you're saying?
In different species, that the tafazzin is the same enzyme for determining other species of cardiolipin other than tetralinolale.
Right.
And if the structural aspects of the mitochondrial membrane
yes.
is one of the determinants,
yes.
can you match those two?
I understand the question, yes. Okay. So the answer is yes. And that is actually a good point. I'm glad you're bringing this up because, it doesn't matter. No, this is, backwards. Yes, here. So this is a good point. Because if you look at the cardiolipin composition in the context of that model, then this goes actually back to the mathematical model that I presented here several years ago. So then the question is, what are the determinants of the molecular species composition of cardiolipin? There are two determinants. One is the overall fatty acid composition. That means it's the fatty acids we are dealing with in the first place. Like, for instance, in yeast, it's 16O, 16-1, 18O, 18-1. That's pretty much it, right? In humans, it's many more fatty acids. So it's different in each organism. So that's one determinant, number one. The second one is the degree of bending, right? How much bending do you need? What kind of non-bilayer structures you have? Obviously, this is speculative. I admit that. But if you look at the morphology of mitochondria in different tissues, they are different. So it's at least conceivable that such forces will drive the difference in molecular species composition.
Michael, interesting hypothesis, and I want to ask you, can you speculate what other forces besides calcium can trigger this transaction between these two phases, hexagonal and bilayer, in living cells, like a pH gradient or something?
Right. So I think it could be as simple as lipid composition itself, right? Because we shouldn't get hung up on the hexagonal phase, because you saw also activation of transisolation in the micellar phase, right? And that was simply driven by an increase in the fraction of LPC versus PC. That was the simplest system. So of course in a cell it's much more complex. You have so many lipids there, and if you have the right composition in a specific domain, it could form a non-bilayer phase automatically without any other changes. That is one possibility. The second possibility is what I am alluding to here as a bending force. Obviously, I'm talking about some external force, and that is, of course, has something to do with the overall membrane dynamics. You know that mitochondria have to fizz and fuse, they have to become smaller, they have to become bigger, they have to do all kinds of things. Who drives that? I don't know, probably proteins, right? I mean, I'm sure there is some sort of a protein involvement. You would not be able to see this kind of intricate morphology that you see in mitochondria in the presence of lipids only. It's widely appreciated that this is a phenomenon that is primarily determined by proteins. But what we have forgotten, meanwhile, while accepting this, of course, as the main factor, what we have forgotten is that these kind of curvatures put lipids under enormous stress, right? Because the packing order gets all messed up, and it becomes just difficult to do it. Now we have to think, sort of go back and say, okay, so what happens if we don't have tafazzin? Well, we have Barth syndrome, obviously. So it's compatible with life, right? I mean, so it's not something that is absolutely essential to have a living organism. But it is something that makes certain aspects of this intracellular function of mitochondria smoother. That's all.
So I had to reach back about 20 years ago because I did my thesis on on mitochondria and calcium. So all of a sudden I had a few things that I remembered. One was, have you ever looked at Carmen Manella, his work on mitochondrial imaging? I think it's called electron tomography.
Yes, I have, yes. Mm-hmm.
So he shows tubes that go through the mitochondria, so they would have a really high curvature instead of
exactly. Yes.
So what you
yes.
see from EM is not quite what's going on there. And the other thing I remembered was about mitochondrial dynamic swelling from potassium. When calcium is released in mitochondria, it causes the mitochondria to swell. And the theory 20 years ago was that when the mitochondria swelled, it exposed more like enzyme to the solution and ended up making more ATP. But that would cause a dynamic change in the membrane, that kind of swelling that happens, like either from the permeability transition, say. But that's something to think about. Like maybe that's the change that you're looking at. And that could be triggered by calcium. I don't know if the amount of calcium that gets released in the mitochondria is actually relevant to the system.
No, I mean, yeah, we discussed that. Yeah,
high,
I think.
Right.
Right. Yeah.
But
so there's many factors that can drive non-bilayer, formation of small non-bilayer domains. Now, we should not forget, actually, that back in the 1980s, Ben de Cruyff and other people have looked at the phase state of mitochondrial phospholipids specifically, and that sort of this issue was settled back then as being primarily in the bilayer phase. Of course, if you have a small non-bilayer phase, with the NMR technology they had back then, they could not detect that. We are hopeful that now we will, with a powerful NMR machine and using magic angle spinning, we will be able to see that. So we are trying to do that, but that is, of course, requires fine-tuning of that technology.
Okay, we'll stop the questions there because